This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 31, recorded October 20th, 2011. Hello, everyone. I am Vincent Racaniello, and with me is the inimitable Dickson Despommier.
The undominable.
Maybe that's what I was looking for.
Or the abdominal.
Dixon, I haven't seen
Hello,
you
Vince.
for TWiP in ages.
I know. We have been ships crossing each other's paths in the night.
Well,
No, really, we've been both very busy, and I've been unable to make my appearances on TWiV, and I feel like I'm suffering from withdrawal symptoms from TWiP. So we need to get back on track, and here we are at another session.
Well, the last time we recorded was well over a month ago with,
Yep.
let's see, the man's name. He's at Pfizer.
Charles?
Chuck Knirsch.
That's right.
That was number 30.
Yep.
And to be honest, Dixon and I have been trying to get another guest. In the meanwhile, it's proven difficult.
Yeah, he's on this floor down below where we
Yeah,
are.
he's in this building, but we'll get him. But in the meantime, a blockbuster paper came out yesterday,
Absolutely.
and we have to talk about it. Plus, we have lots of backed up email,
We do.
so we'll do a TWiP.
Excellent.
And then I'm off to Brazil, and you're off to China.
That's right. But we'll be back.
And maybe if our planes crash, we'll never be back.
No, but if we bring something back with us, we can talk about that on the next TWiP.
You're going to bring a parasite back with you?
I don't think so. I'm going to try not to.
Not that you know of.
Not that I know
It
of.
could happen.
It could.
We just had the wonderful pleasure of seeing your son,
That's true. It's a
who was
pleasure.
a
seeing my son.
voiceover expert,
He is.
and he did some intros
He did.
for all three podcasts, and
That's
maybe
right.
this one will play with one of them. But if in the future you hear various famous people introducing TWiV, TWiP, and TWIM, you'll know who it is.
Correct. All the way from Rodney Dangerfield to Bill Clinton.
Did he do a Rodney?
He did.
He did a Dixon. That was my favorite.
He did Dickson. Come on.
All right, today's paper
Ah,
came out this week in the New England Journal of Medicine. I don't know if you know anything about this, Dixon. It's something called malaria.
I've heard of it.
We did 3 episodes
We did.
on malaria. I have your textbook here.
We need to do a lot more on malaria. It's
We do?
the world's most important infectious disease, bar none. It's true.
Okay. So Bill Gates is very interested in malaria.
He is.
He would like to put his name on conquering it, right?
He would love to do that.
So the name of this paper is First Results of a Phase 3 Trial of RTS-S/AS1 Malaria Vaccine.
Just rolls off the tongue, doesn't it, Vince?
It's quite a name. RTS-S/AS01 Malaria Vaccine in African Children.
Right.
And this is being touted in the press as an amazing result.
Correct.
So let's talk about this, can we?
Absolutely.
And I have a lot of questions for you.
Okay.
Let's talk about this vaccine.
Yes. Or
But
any vaccine.
According to this article, Dixon, it says that malaria occurs in approximately 225 million people worldwide, and 780,000 die.
That's a conservative number.
And those are
in most
mostly
cases.
African kids who
That's
die.
right. That's right.
So that's a problem for the future of Africa, right? Because they never grow up to be productive adults.
That's right.
Which plasmodium is it that does most of the damage?
It's mostly Plasmodium falciparum.
Falciparum or falciparum? You remember this discussion we
I
had?
do. You say storm door and I say potatoes.
You know, we make a good pair. We should do radio.
We are doing
Oh,
it.
I forgot.
I
Sorry.
consider this radio.
It is radio.
That's right.
Plasmodium falciparum
Or falciparum.
or falciparum, and that is what this vaccine is against,
That's
right?
right.
So let's talk a little bit about the vaccine itself. It's been out for a long time.
It has. In fact, it's a well-known antigenic construct that was first discovered at NYU in New York City.
So this targets the Circumsporozoite protein. What is a Circumsporozoite? Can I see a picture of it? That would help.
Well, you can see a sporozoite, that's for sure.
This goes around it, I bet.
It does.
Where's the sporozoite?
Well, you've got some pictures of them here in the life cycle drawing. Okay. But you've also got some pictures here. sporozoite stage of
Remind
this
us, yes.
parasite is the stage that lives in these large structures on the surface of the stomach wall of female Anopheline mosquitoes. And they produce thousands in each of these large structures called the oocysts.
Mm-hmm.
In fact, the parasites can actually get out of the oocysts without the oocysts rupturing, so they can continuously produce for quite a while. And where do they go? They go into the hemocoel of the mosquito. The hemocoel is like the blood space, okay? Most insects, in fact, no insects have a complete circulatory system. Their organs are bathed in a fluid called hemolymph.
Mm-hmm.
And so the sporozoites accumulate in the hemolymph. And because the insects have open-chambered hearts, which circulate the hemolymph around all the organs, eventually the sporozoites are just sort of tag along with all the other elements in the hemolymph. There are some cells in the hemolymph as well, which aid the mosquito or bug in resisting diseases of various sorts, but not this one, unfortunately. So eventually these sporozoites find their way into the salivary glands of the female Anopheles mosquitoes and into the hypostyle, which is the apparatus that it uses to suck your blood through.
Okay.
So that the moment that this female mosquito now encounters another person, and you say, what do you mean another person? Well, in order to acquire malaria to begin with, the uninitiated virgin female mosquito had to take blood
From an infected person.
Yeah, and she's doing that to sequester the protein from the red cells,
right,
so that she can put that protein to good use and make eggs with it.
Because of course malaria does not exist in mosquitoes naturally, right?
You can't. They call it horizontal or vertical transmission. It can't vertically transmit from the female mosquito to the eggs to the pupae and the larvae.
She has to acquire it.
She has to acquire it every time, so she has to bite twice.
When she does, she acquires sporozoites.
No.
What does she acquire?
She acquires the gametocytes.
Okay.
They're called gametocytes, but they're pre-sex cells. Right?
Okay.
They're both haploid, and they're either male or female. And inside the gut of the mosquito, these things differentiate into
and become sporozoites, which then
no. They don't become sporozoites. They then have to mate. They have to become diploid first.
Okay.
And once they do that, they form something called the ookinet. And the ookinet now is a diploid organism. It actually migrates in between the gut cells and lodges in the membrane.
Right.
It then transforms into the oocyst.
Right.
And inside the oocyst, it undergoes nuclear reduction first to produce haploid cells, and then the haploid cells differentiate into sporozoites.
And those are what they pass on to us.
That's the infectious stage.
And that is why the vaccine is targeting.
Correct.
So the circumsporozoite protein must be around
the
sporozoite.
It is, and it's an interesting protein. It was discovered through a collaborative effort at NYU with the well-known malariologists, the Nussensweigs, and also with Dr. Gerald Vandenberg, who made the original discovery of if you irradiate sporozoites and then inject them into a mouse model for malaria, in this case it was Plasmodium burgii, the sporozoites are attenuated. They will not complete the life cycle.
Right.
But what they will do during their living stage is impart immunity to an incoming life cycle. So it was known that the sporozoites contain functional antigens, which you could then use as a vaccine. And then the Nussensweigs discovered that the Circumsporozoite Antigen, which is surrounding the entire sporozoite, is a repeated peptide. I think if I recall correctly, it has the 36 or so repeated tetramers of a particular.
Called a NANP repeat.
NANP.
Which means any amino acid, alanine, any amino acid, proline.
Bingo. I knew that.
It's okay. And so by targeting this protein, you prevent yourself from being infected.
Well, the data on that is that if you go into native populations in Africa that have resisted this infection for almost all their lives, they contain antibodies which will kill the sporozoites. And it reacts specifically with this Circumsporozoite antigen.
So there are some individuals who are naturally resistant.
Yes, but I'm talking about acquired resistance now.
Yeah, it's
repeated infections. Okay, but the point is that when they tried to take this circumsporozoite protein and immunize with it, they got very low or no resistances. Although they knew it was the right antigen, they weren't giving it correctly, perhaps. So they started to look at whether it was a T cell or a B cell response, and they got involved in the immunity to malaria, which is very fleeting, by the way. Malaria, if you visit a malaria area and you live there for 5 or 6 years and you acquire malaria 3 or 4 times, which is likely, you end up resisting incoming infections. But the moment you leave Africa or wherever you were living and you come back 2 years later, you're as naive as you were when you first moved.
So the immunity wanes.
It wanes.
Memory is not
very,
robust.
very low and fleeting amounts of
Hopefully
memory.
the vaccine will be better than natural infection.
Well, there's some
We'll
hints.
see.
There's some hints that the people that were immunized with the vaccine 2 years out you couldn't demonstrate any antibodies in their serum.
So according to a paper I'm looking at where they first, among the early papers where they characterize this vaccine, they say both antibodies, circumsporozoite antibodies, and T cells are implicated in protection.
Right. That's right.
So the antibodies would presumably bind the circumsporozoite and somehow target it for destruction.
Sort of a antibody-mediated cell cytotoxicity reaction. That's what they were looking at.
Could be lysed by complement
right.
or taken up by phagocytic cells.
Sure.
And then the T cells would actually lyse the circumsporozoite directly.
Perhaps.
I understand that liver cells have this antigen on their surface. Is that correct?
Well, they've got a receptor for the sporozoite. Actually, the receptor is in the sinusoidal vessels that are lining the hexamered arranged parenchymal cells in the liver. Okay, there are a series of hexagons in the liver, and at each junction you've got a sinusoid.
Okay, it's in the cytoplasm of the hepatocyte. Okay.
So as the mosquito injects the sporozoites into your blood, as soon as they pass into that zone in the liver, which is a large organ, as you know, the receptor on the surface of the sinusoid and the sporozoite form a ligand.
Okay.
And the next thing you know, you've got the sporozoite penetrating through the sinusoid into the cuboidal cells that are the parenchymal cells.
Now, a little bit of the history of this vaccine. The initial candidate constructs, that is targeting only the central repeat region of the circumspore. We're going to call it the CS protein from now on, so we'll save some time.
That's right.
Failed to provide substantial protection.
Correct.
So then they decided they need to modify the vaccine to recruit better T cells. And so actually the groups involved have been the Walter Reed Army Institute. Does that make sense?
Absolutely.
They have always been involved in.
Well, they say that during the Second World War events, more troops on both sides of the fence, so to speak, from the United States and Japan died from malaria than from bullets. So that shows you how important malaria has been.
Also, GlaxoSmithKline Biologicals are heavily involved, and money has been given by the Gates Foundation.
You bet.
Who would like to put their impact on some major disease.
That's true. And there's another group in Seattle called PATH, which is also involved in this process as well.
So then they made a new construct with a larger segment of the CS protein.
Yep.
Again, from P. falciparum.
Yep. Expressed in yeast.
Let's see. This one is covalently bound to the hepatitis. Yes, you're right, Dickson. Expressed in yeast, but the key difference here, it was fused with the hepatitis B virus surface antigen.
Now, why would they do that, Vince? Now, this is not This Week in Virology. This is This Week in Parasitism. Why would they take a virus construct and tag it onto a malaria antigen?
Well,
what's the advantage of that?
The virion of Hep B is a wonderful adjuvant. It's a natural adjuvant. It's a repeated structure, and it's beautifully presented to the immune system.
All right.
So by incorporating, maybe the CS antigen they picked is not a very good antigen, doesn't induce inflammation, which you need for a good immune response.
Okay.
But by coupling it to a wonderful antigen, which we know the Hep B surface particle to be when it assembles into this beautiful virion.
And there's a vaccine using it.
And there is a vaccine which consists of just the Hep B surface antigen.
So they've jumped through those hoops in order to test it out in people. So no, that's not toxic.
So you just express the surface antigen of Hep B and it makes particles. It makes empty particles.
Wow.
Those are highly immunogenic.
Okay.
You can express those in yeast or in insect cells, and that's the vaccine that we actually use today.
Terrific.
So you can just fuse your protein antigen onto the Hep B surface antigen and make a particle that now has these malaria candidates, and that is now found to be much better at generating an immune response against the CS protein.
How about that?
And it says here, these Hep B particles favor antigen presentation to the T cell compartment.
That's great. Now, I wanted to back up just a minute here, Vince, and convince you that the field of malariology is an old field, okay? In fact, some of the first Nobel Prizes ever given were given for work on malaria. And, they gave out a prize for discovering that malaria is transmitted from animal to animal by mosquitoes, okay? And that was Sir Ronald Ross when he was just Ronald Ross. But he worked in canaries, and we discussed this when we were discussing the biology of malaria infections. Then another guy got it for discovering that malaria has sex in the stomach lining of the mosquitoes. That's another Nobel Prize. But nobody got a Nobel Prize for studying how people become immune to malaria, because people don't become immune to malaria. That's the big problem. And it turns out the reason why they don't, for the most part, is the fact that the malaria parasite undergoes antigenic variation.
Really?
It does.
Very much like the
trypanosomes.
Wow. And also influenza virus.
All the good ones know how to avoid us no matter what. So there's no variation on the antigen on the sporozoid.
Right.
None.
So how does it get around?
So that's the beauty.
Immunity.
Well, it's in the blood so fast and gets to the liver so quickly that there's almost no time for the immune system to encounter it and to defeat it.
Dickson, it sounds to me like this is a tough vaccine problem to crack.
Say another one that's tough. HIV. Say another one that's tough.
All the easy ones have been done,
Tuberculosis.
Right?
Say another one that, of course, all the easy ones have been done. So this is a toughie. So this is big news now.
So Dickson, if something is tough, should we just not bother with it?
Well, a lot of people did say that. The best way to avoid malaria is to stay away from it.
I suppose if you have money, you can just pursue it, which we're fortunate that Gates has stepped in here.
Well, sure. Once you see what malaria does to a country, when you see what it does to the people that live there, what it does to the economies, if you're ever going to watch this world come up to snuff with regards to equality, you have to solve these basic infectious disease problems first. And that's true for HIV and tuberculosis as well.
So this vaccine candidate that we've talked about now, it's a different CS protein fused to Hep B, is also given with 2 adjuvants.
Aha. What's an adjuvant, Vince?
An adjuvant is a chemical.
Is that somebody who works at the university only part-time?
I thought that was adjutant. You know what an adjutant is?
You'll tell me.
I don't know what it is.
We're going to get mail on this. I know we will.
Well, I'm going to look it up. An adjutant, fortunately I have a computer, is a military officer who acts as an administrative assistant to a senior officer.
There you go.
It's also a bird.
Oh, really?
It's a large black and white stalk.
An adjutant.
Found in India.
Had no idea.
Southeast Asia, but we digress.
We do.
An adjuvant is a chemical added to a vaccine to produce a better immune response. It does
Name some.
Well,
I mean, I'm not quizzing you.
I'm going to name one that's only used in experimental animals, in which most people would know. Freund's adjuvant.
Correct.
Can be either incomplete or complete, which you add killed mycobacteria.
That's complete.
It's a wonder. It's got oil. And it's got bacteria that stimulate the immune response.
And you emulsify your antigens in it and inject
it. When you inject it in the rabbit, it causes inflammation.
Sure.
It causes recruitment of immune cells and secretion of cytokines, and that helps make for a better immune response.
It makes granulomas.
Granulomas.
I used to use it all the time in my research.
So if you want, I'll send you to my immunology lecture to learn more about that. But here in the U.S., we include adjuvants in just a few vaccines. The Hep B is actually one of them, and the human papillomavirus vaccines to boost their...
What are they using there?
Those adjuvants are aluminum sulfate based.
Called?
Well,
alum?
What is alum, Dickson?
Alum. That's an aluminum-based adjuvant. Sort of an inert material that holds the antigen in place in the tissues until the cells can come and
so some of the common components are aluminum, salts,
right.
Oils of various sorts.
Right.
We don't use the bacteria in people, the mycobacteria.
No, the oils are lanolin, as I recall.
But the malaria vaccine uses 2 different kinds of adjuvants. They're called AS01 and AS02. These have been approved for use in humans, and they're used in other countries. And the components include a chemical called MPL, which is a monophospholipid A molecule.
Right.
It contains a detoxified form of lipopolysaccharide, which is a bacterial outer membrane protein.
Highly antigenic.
It's highly antigenic and highly immunostimulatory, highly inflammatory.
For cellular responses, not
well, what this does,
responses.
I'll tell you what this does, Dickson. This LPS hits a receptor on the cell surface called TLR4, toll-like receptor 4, and it induces innate responses, recruits cells, they make cytokines, and eventually the adaptive immune response gets involved. Okay. So when you have good inflammation, you get a good adaptive response, which means antibodies and T cells. So the LPS is a natural adjuvant, which they've incorporated here.
Clever.
Other things in this, a natural saponin molecule purified from
saponin, depending
Sorry.
on
I wasn't well educated.
Saponin, you said that a different way, Vince.
I said saponin.
That's right.
But who's to say what is the right way to say it?
It doesn't matter.
Saponin?
As long as you spell it right.
Did you say saponin?
No, I said saponin.
Saponin, S-A-P-O-N-I-N, which is purified from the bark of the South American tree. I'm not going to pronounce it. Q. saponaria.
That's right.
It also contains an oil and water emulsion and a liposomal suspension. So these are all additives that have been shown to induce inflammation, but are also safe to use in people.
Of course.
It has to be safe.
It does.
And contrary to popular belief, they're not dangerous. They have been tested. Yeah. Now, they will make you a little bit more sore at the injection site.
A small price to pay if it works.
Exactly. This is nothing. And it's been shown in many trials that, in fact, you get more side effects like soreness and fever and that sort of thing with an adjuvant. But if you get a better immune response and you're protected against...
I'll take it.
I will take it, too.
Give me 2 of those.
Do you think the 700,000 plus kids who died of malaria would take it?
Of course they would.
If they could. So that is the vaccine, and it has been already through phase 1 and 2.
Which is?
Phase 1 is a safety trial.
Right.
Small numbers of people.
Right.
You put a single dose in, typically, or it can be modified to see if there are any nasty side effects.
Right, like fever or lasting muscle pain.
Or death.
Because
if people die, you suspend the trial. Yeah. And you have to tell people when you sign up for these.
Here are the possibilities.
These are the possibilities. Right. But you have already been in extensive preclinical.
In animals.
That's right. And if your animals have died, they will never go into a human.
That is
so correct. They've been safe in animals. But as we all know, non-human animals are not human animals.
There you go. I couldn't have said it better myself.
So penetrating.
Yes. And so logical.
And what is a phase 2 trial?
A larger number of people.
And what's the goal of a phase 2 trial?
I guess to show how to go about doing a phase 3 trial.
I sense that you are...
not big on clinical trials here, Vince. I know what phase 3 is because that's where we're at now. But phase 2, I think, is a small version of phase 3.
The main goal of a phase 2 trial is to identify the therapeutic efficacy. They're usually single-arm studies, but sometimes they can be multi-arm. So you want to know if they have any effect.
How would you do that?
Well, you have to do a little protective study.
Yeah, but how would you show the protection exists unless you give them the infection?
Well, you can't infect people, obviously.
Yes, you can. Yeah, in fact, that's what they did.
They infected them?
They did.
All right, we're going to see. But a phase 3 is a larger one. Sometimes you look for different doses.
Phase 3 is out in the field.
Out in the field.
Phase 2 is not out in the field.
Well, if you're looking at an infectious agent, let's say it's an HIV vaccine, you can't infect people. You just let them go about their normal lives and let them become infected.
But malaria is different.
You can infect people?
You can.
You're going to get a lot of email about this, Dickson.
No, Vince, it happens. And in fact, they gave the volunteers, these are volunteers, they were quizzed ahead of time as to whether they had any doubts about any of this. And they let mosquitoes feed on them. And they were using a chloroquine-sensitive Plasmodium falciparum infection. It's the only way that you can test this vaccine. So these are volunteered people who got the vaccine and who then had to be challenged either after 1 immunization, 2 immunizations, or 3 immunizations with one of 2 different adjuvants. That's the phase 2 trials. Tough. And you have to have enough people to make sure that this thing has statistical significance. So they had I think 12 or 13 people in each of these groups.
All right, so we've been through phase 1 and 2.
That's right. So phase 2 said,
What did the results of the phase 2 say?
It said one of the constructs with one of the adjuvants gave a 50% protection.
Mm-hmm.
More than 50%, actually. It was higher than that. 2 of them didn't give any protection. But the third trial gave protection. It meant that you were now ready for clinical trials.
Yeah, so this one, RTS-S/A01, was the superior formulation.
That's right. So now we can take it in the field and see whether it protects little babies.
Dickson is looking at some birds. What are they?
Those are shags. They're cormorants.
Really? I thought they were geese.
No. Those are cormorants.
Where are they going? To dinner?
They're going to go find fish.
Are they talking to each other?
I hope so.
So let's make sure everyone understands what phase 2 does.
Right.
So basically, if you fail a phase 1, you don't go any further.
No.
But if it's safe in phase 1, you move to phase 3, which gives you some indication on efficacy. Right. All right. And then if you fail a phase 2, if it's not efficacious, you don't go into 3, which is a much bigger study and can involve multiple doses, right?
Right. But with HIV, you can't do a phase 2 study because you can't cure the infection.
Well, you can prevent it.
No, but you can't test the vaccine by giving them an HIV infection after they take the vaccine.
But what you do with HIV is that you have an at-risk population.
I know.
You immunize some and you give others placebo.
You're right.
And then in a year or 2 years, you look at the rate of HIV in both groups.
That's a phase 3 study for malaria.
Yeah, you can't do that in phase 2.
If you have HIV virus, you can't cure it. So
you use different people, Dickson, obviously.
No, I know that. But what I'm trying to say is that with malaria, you can do a phase 2 study because you can cure it. So you can do it on a small group of people.
Not cure, but prevent infection, right?
Well, they were...
Well, it depends what the target population is.
Even if the vaccine doesn't work, you still have to be able to get rid of the infection, you see. So you have to assume that it won't work. And if it doesn't work, what are you going to do about it? That's in phase 2 for malaria. So with malaria volunteers, they let themselves be fed on by infected mosquitoes after they got vaccinated in a certain way, okay? And then they measured their fever and the appearance of the parasites in the bloodstream. Mm-hmm. And the moment they saw parasites, they cured them with chloroquine. And they said, this was not prevented. This infection wasn't prevented. So that vaccine trial didn't work.
So that is necessary because we have a drug that can prevent.
Correct.
Right, we have chloroquine. You can't deny people something that exists. That's unethical.
Exactly right.
Now, with HIV...
That's different.
You have to counsel people to avoid risky behavior. But you also have to bring them in periodically and check them if they've become infected. And if they do you know what you do? You give them antivirals, of course. Triple therapy, because you can't let them keep going.
That's correct.
Immediately treat them.
That's correct. But you can't purposefully infect them.
No.
Whereas in malaria, you can.
Why?
Because we have a drug for the parasites, so that even if you get an infection...
Oh, all right. So now I finally understand what you were saying, because you can cure of malaria, but HIV you're stuck with because it integrates, and we don't know how to get rid of the integrated copies. Okay. I was just not understanding you.
But Vincent,
it's my fault.
No, it's my fault.
No,
it's my fault.
I'm in a generous mood today. It's my fault. But don't depend on it every day.
You know what? He's going to edit that out. That's okay.
No, I'm not going to edit it out. It's a click and clack, right?
It's click and clack.
So we move to a phase 3, which is what we have here, and it's called First Results of a Phase 3 trial.
Thousands of people now.
How many thousands?
17, as I recall.
All right, let's take a look. We got the paper right in front of us, Dickson.
But I don't see it, and you do.
From March, turn it around. Oops. From March 2009, can you see, or is it too small?
I have to put my glasses on now.
To January 2011, they enrolled 15,460 children to one of 3 study groups.
Right.
They used comparator vaccines, rabies vaccine for children of 5 to 17 months of age, meningococcal vaccine, or meningococcal vaccine. So they did 3 vaccinations.
2 of which were guaranteed not to protect against malaria.
Yeah.
So those were the controls.
But they would protect against something else.
Rabies and meningococcus.
So it's not bad, right?
What were they? They were showing that just by immunization alone wasn't enough. You had to have the right antigen present.
Right.
Those were the controls.
Well, you can get inflammation as a consequence.
Yeah, that's right.
Immunization, and you want to control for that. But it's interesting that they use 2 different vaccines.
Yes, because they were going to give it to them anyway.
So they did passive surveillance for malaria from the time of the first dose until the end of the study.
Right.
They gave a variety of doses, which we don't need to go into here. And then
it was a total of 3 injections for maximum protection.
3 injections.
By the way,
how were they given, Dickson?
Intramuscularly. But this is...
Did they use needles?
They did. Of course they did. No, they could have used an air gun.
Yeah.
Could have used an air gun, but that only gives subcutaneous injection. But the real issue here is 3 injections, Vince. Even if it works.
That's hard, right?
Tough getting the same person to come back 2 more times.
How spaced out were the 3 injections? At least a month, I would guess.
Yes, it was. In fact, it was.
I don't know the exact number.
Day 0, 1 week, and a month later, I think there was a 3rd injection, if I recall right.
I don't know. Let's see here.
It says
For
everything.
details, see the supplementary appendix.
You're not going to believe the supplementary appendix.
This is probably huge, right?
It's 55 pages long.
Yeah, it's got the whole protocol in it.
It's unbelievable. It's like 2 more papers.
Thank
God for the internet.
Tables.
Thank God for the internet.
We'll put a link to this.
Yeah, it's worth it, by
so
the
you
way.
can see it. This is quite a document, yeah.
You know what? The same document could be seen with the big HIV study that failed about 3 years ago.
Yeah.
And
of course, those are huge.
So that's why this is so important, because it succeeded. Here's one of those 3 big organisms that they've identified through the WHO as needs to be conquered: tuberculosis, AIDS, and malaria. At least we have some hope for malaria now.
But Dickson, do you know, first of all, this was all in Africa.
That's correct.
All right.
So that's going to be different wherever else you go.
Right, but...
And it's only for falciparum. It doesn't even cross-react with the others.
The thing that's amazing to me is that they enrolled a lot of kids.
They're only kids. They're only worried about kids, Vince. Why?
5 to 17 months of age.
Why is that?
Wow. And what are the older ones? 6 to 12 weeks of age. 6 to 12 weeks of age.
I know.
That's amazing.
I know.
Anyway, so the all African and all very young kids.
And it was all West Africa. It wasn't even East Africa. It was all West Africa.
Actually, in this supplementary appendix, it tells you exactly.
The map of Africa is
where?
One of those
Here,
tables.
tell me these countries. We got Burkina Faso.
That's West Africa.
Gabon.
That's West Africa.
Ghana.
West Africa.
Kenya.
Nope, that's East Africa.
Malawi. Mozambique, Tanzania.
Tanzania.
That's the range of transmission, they say, across sub-Saharan
Yeah, but that's not
Africa.
where the studies were done. If you go to the map,
back to the paper or to the supplementary?
Supplementary.
Here it is. Here's supplementary. Each child received 3 doses, okay?
Right.
And the controls we've talked about.
That's a massive study for 3 doses, Vince, for 15,000 some odd kids, to keep track of every one of those kids to make sure. You know how much work that is? That's an enormous amount of work.
I'd say they don't make it obvious
but it's
how much time. Oh, of course, it's a lot of work, yes.
And it requires teams of coordination and preparing the antigens and make sure that everything is sterile and making sure the needles are fine.
And they monitor everyone for adverse effects.
It's incredible.
Long time.
It's incredible. And you know what? These people all volunteered for this because they realized that they were trying to do some good, and they came back with their kids because they wanted to see this thing to completion.
And then they say to the parents, if your child is sick, bring them back, and we'll see if they're getting malaria.
Exactly.
And then you collect this data, and I guess you can assay them for malaria quite easily. What would you do?
It's not easy, but you can do it. I mean, you can do a PCR. You can do but if you do a PCR, you better make sure it doesn't try to detect. Well, no, the PCR would be fine. Yeah, PCR.
So they did some hematology.
Yep.
Here.
Yep.
Standard microbiology, looking for other pathogens.
Did they use microscopy as a diagnosis? I don't think so.
I'm looking here. Counts by blood smear. P. falciparum counts by blood smear.
Microscopy.
Read by 2 microscopists, one of whom was Dickson Despommier.
No, please don't do that.
No, you don't want to do that?
No,
Did you ever read malaria smears?
Sure. Lots of
Wow, I'm in the presence of a person who's done that.
Oh, of course.
Amazing. Could you still do it?
Absolutely.
Antibodies, too, for circumsporate?
All these pictures in this book, I took these pictures.
They measured antibodies against the circumsporozoite antigen in these children.
Yeah, that's the part that's disappointing, because after 16 months, there is no antibodies.
So that you want to know how that correlates with whether the kids got...
So they immunized them just before the maximum transmission season. There are seasons for transmission in malaria.
Uh-huh.
So you don't give it after the season.
Oh, that makes sense, right?
Yeah, because otherwise there's a big lapse of time before they get exposed again.
Okay, so what are the results? But it's not finished yet, right?
It's not finished, but it's done enough so they can say some good things about this vaccine. The group that was protected maximally.
You know what? That they were monitored for 12 months after the third dose. It's important for us to say that.
Absolutely right.
Okay.
So that got them through at least 2 transmission cycles. And one of the groups had over a 50% reduction in clinical malaria. Okay, clinical malaria. That's the way to put it. They didn't get sick.
That's right. They say during 12 months of follow-up in the first 6,000 children in the older age group, the incidence in the first or only episode of clinical malaria meeting the primary case definition was 55%, showed 55.8% vaccine efficacy.
Right. Means that only 45% got sick.
Out of 6,000 children.
That's great.
How many would have gotten sick otherwise? Let's look at the controls.
That's right, Vince. Well, it's 55% over the normal.
I got a table 1 here with this data. This is quite
almost all
amazing.
of them got sick.
In the controls?
Yeah.
Let's see. Oh, this is a fancy table here. It's so small that I cannot read it. Let's do full screen.
Yeah.
All right, here's our control vaccines. All right, first only episode. Here's the 55% protective efficacy. In the control vaccine, we had 900 people, 80% infection rate.
Right.
In the vaccinated population.
And by the way, when they discover that, they treat them. Okay? So
yeah, of course.
They're going to treat these kids when they become positive.
So in the vaccinated population was a 43% infection rate. So it's about half of without vaccination.
Exactly.
That's where you get the number of roughly 50% protection, right?
That's right.
So that's why this is exciting, because it's the first time that we have seen such strong protection, right?
It's very exciting. And by the way, it's worth mentioning that there was another program to prevent malaria, not to immunize, but rather to prevent.
How? With what?
With bed nets.
Oh, yes. How did that work out?
And we have a gentleman on our staff here at Columbia University. His name is Jeffrey Sachs. He's the leader or the director of the Earth Institute on our downtown campus. And his main reason for existing for many years was to make sure that enough money was generated. It's $10 a bed net, by the way. That's not a trivial number when you consider how many people are trying to give them out to. He reached the 50% mark in bed nets. Remarkably, Vince, the rate of malaria in many countries dropped by 50% after that.
Yeah, the bed net is a low-tech but effective method.
Correct.
Right?
It has to be insecticide impregnated. Okay?
It does.
It does. Once a year. They do that once a year. So they take their bed nets in and they wash them and then they dip them in an insecticide and then they dry them. That's good for about a year. It's a residual insecticide. I presume it's DDT, but I'm not sure. So Jeffrey Sachs has been a champion of that technology. So if you couple that with the vaccine,
you could really reduce it.
So there's a group that's now advocating eradication again. We didn't hear that word since 1960s.
Of malaria?
Yes.
That's kind of a bit much, isn't it?
That's radical. There's a group that wrote an essay, and I got it up on PubMed. It was in 2011, that said we are now back at the point of being able to consider eradicating malaria based on these double-pronged approaches of vaccines plus bed nets. The drugs were the third one, of course, and that didn't work at all because the organisms became resistant.
Yeah.
But these two other approaches could very well combine together to reduce it below the transmission level, because you need infected people to give it to the mosquitoes so that they can give it to other people, because they don't inherit this from their mothers, the mosquitoes. So if you reduce the numbers below a certain level, back in I guess it was the 1950s and 60s, there was a guy by the name of, I believe it was McGregor, but somebody will correct me, of course, that did a mathematical modeling of how many people do you need to have infected in order to maintain transmission in a community. And it was a remarkably high number. It was higher than you thought. So if you could reduce malaria below a certain limit, you could eradicate it. Okay, so that's why they got into this eradication mode back in the 60s. And that's just about when chloroquine resistance first started to come up.
They do note here, Dickson, that there were some serious side effects.
Did they?
Yeah.
Name them.
Let's see. in the older age category. Serious events were reported. At least one was considered to be related to the vaccine. In 11 children, 7 seizures, 3 episodes of pyrexia, 1 episode of myositis, and 1 injection site reaction, and 1 seizure in another group. So no death, but seizure is pretty serious stuff.
Yep, it is.
It's 11 out of 6,000.
Vince, that's probably lower than you'd get from aspirin therapy. Probably, I mean, a lot less.
They also say they couldn't see any reduction in malaria deaths. So there were a number of deaths caused by malaria during this period.
Overwhelming infection.
And they weren't a lot of them. So statistically, it was difficult for them. Malaria-specific morbidity was only, 10 out of 150 kids died during the trial.
Right.
You would expect in this country of 6,000 kids, a lot of them are just going to die malnutrition or accidents or whatever. 10 of those were from malaria only. And so they couldn't see an effect of the vaccine on that because the number is too small.
Right.
But it's basically 50% protection after 6,000 kids have been evaluated.
That's the best result ever so far with any vaccine for malaria.
And so when the other children are evaluated, what if it's still 50%? Would they go forward with this and start immunizing?
They would.
Or do they need to tinker with it some more?
Well, someone from the Gates Foundation was quoted as saying that we wished it had been 100%. Because they asked the Gates Foundation whether they would fund the Glaxo study to make sure that there's enough vaccine for millions of people, not just on thousands of people.
Yeah.
And they sort of hedged their bets on that one.
It's an interesting question, right?
It is. But the Glaxo company, GlaxoSmithKline, said that we will offer this vaccine at a fraction above the actual cost of making the vaccine.
Good.
So they're going to reduce all of their input costs that they would have for other drugs.
But it's not just the vaccine cost, it's getting it injected, right?
No kidding. And it's
expensive.
Follow-up, and it's, what if you had to have an influenza vaccine 3 times before you were finished with the vaccination procedures?
The repeat is tough, but...
Very tough.
You have people on the ground whose job it is to do this and say, hey, look, we can perhaps help you with this disease that you've lived with all your life as an adult, and your kids are going to get it. So
the amazing thing is when you compare malaria to other infectious diseases, like, say, for instance, plague, right? And when plague hit Europe the first time, it wiped out, like, say, 30 or 40% of the population. And the next time it was like 80 years later, those people who had the genes to resist naturally,
right,
then repopulated Europe.
Sure.
And the next time the wave of infection came through, it was only like 15% death rate. And then the next time it came through, it was only like 3% death rate. So every time you see a selection towards resistance, which equals a certain genotype, with malaria, that doesn't happen. You know what the selection pressure was for?
What?
What do you think? Sickle cell.
Ah, yes.
And
of course.
Thalassemia. So these are blood dyscrasias that have a bad effect on the growth of the parasite inside the red cells. Right. The resistance genes for malaria probably don't exist because it's such a complicated infection. So instead of selecting people resistant to malaria, they selected people in favor of these balanced polymorphisms of these genetics that equals another disease that's less severe in its heterozygous form, which then still offers some protection against malaria. It's a crazy world when you look at all the interactions. And by the way, you must also recall that Africa was where human beings first arose. So we have been playing with these infections ever since we arose out of the dusty plains of East Africa. It's a sobering thought, to be honest with you. And the best way to avoid infection is to move.
Yeah, well,
now, maybe that
not everyone can do that.
No, but maybe that's what caused us to migrate to begin with. You say, why did people migrate out of Africa?
You think we realized it back then?
Well,
I don't know.
There was a time, Vince, when we were down to 400,000 individuals.
Could we even talk and say let's get out of here? I don't know. I don't know if we had the cognitive abilities. I have no idea what it was like.
We had the ability to talk because our voice boxes are unchanged since then, whereas Neanderthals didn't have the ability to talk like that, right? But the point is that we've only been out of Africa for about 70,000 years. That's all. We
I
stayed
suppose we could say, listen, guys, this disease, whatever it is too much. Let's see what's on the other side of the fence.
Or, I'm tired of having 20 kids and only 2 surviving.
Yeah. So the long-term duration of the antibody, they say, remains to be determined here.
Correct.
When they do the rest of the study. And then they'll tell us how the levels, and I guess these kids who were immunized and didn't get infected in these in this first year of surveillance, you'll see what happens to them, right?
Well, yeah, if they don't get boosted, then that's true.
You follow them up for a few years and see how long the protection is. Yeah. And if you need to, you boost them at some point, right?
Sure.
Because if you start to see breakdown of immunity, and so that's essential to follow these 15,000 kids for a while. It is,
but here's another monkey wrench to throw into this thing too, Vince.
I thought the HIV one was amazing.
Well,
this is kids.
The worst manifestation of Plasmodium falciparum infection is called cerebral malaria.
Mm-hmm.
That doesn't mean it makes you think better. What it really does is it affects the way you think in an adverse way.
Yeah.
So you go into a coma and you survive it. And when you emerge from the coma, you're a little less smart than you were before. And so what they've shown in Kenya, at least, is that if you survive 6 or 7 seasons worth of this infection at a cerebral level, you're a lot less smart than you were when you started your experience with this infection. So even if the vaccine saves your life, but doesn't prevent this form of the malaria,
right,
it's still going to take its toll on people.
Yeah, that's right.
Right? What you want to do is prevent cerebral malaria in any way you can. And maybe if you have a vaccine that works at 50% level to prevent illness, maybe the illness that they're talking about is this cerebral variety.
Right here I found something on the immunogenicity. The titer of anti-circumsporozoite antibody was low in the 2 study groups. 1 month after the 3rd dose, 99% of children were still positive for antibodies with a low titer. But, they didn't measure T cells.
No.
Maybe they're as important as well.
Could be.
But so this is we still have more to go, but it's very promising, right?
So it is very promising. So my other question then is, wouldn't you rather have a vaccine that prevents infection rather than modifies an existing infection?
Isn't that what this one is doing?
This one is modifying an existing infection because all of the people in the study got infected, but only half of the ones that were infected got sick.
By infected, you mean they were bitten and had sporozoites injected into them. And how are you going to prevent that? You got to get rid of the mosquito, right?
No, but this is a sporozoite antigen, though, you see. In the irradiated antigen study, where they irradiated sporozoites, it's 100%.
100% what?
100% non-infection.
By infection, do you mean invasion of liver cells?
It prevents it 100%.
So this is not 100% preventing that.
But it's not preventing it at all.
But it prevents clinical malaria, right?
But that's different.
I understand.
There were a breakthrough. They call it a breakthrough infection.
I understand.
Okay.
So you're saying you want to completely prevent reproduction of the malaria parasite within the human, right?
If this is a sporozoite antigen, then I want to prevent that sporozoite from succeeding. So what this probably does is as follows. Let's say, how many sporozoites do you think a mosquito injects into a person?
300 or 400?
Maybe more.
1,000?
Perhaps. Let's take it as 1,000. Okay. So what this vaccine, I think, is doing is it's reducing the number of hits to the liver.
Yeah. Okay.
And it's keeping it low enough so that as the parasites start to multiply in your blood at low levels, your immune system can kick in and start fighting that off, and you prevent disease. You don't prevent infection.
That's a good point.
All right. So even if one of these sporozoites gets through, you're going to get infection, but you won't get sick. So what I want to do is prevent 100% of the sporozoites from succeeding. I'd love a vaccine to do that.
Well, that may not be the first generation.
Of course, but we're on the right road here.
But even this vaccine would be, if it pans out in the rest of the study, it would still be useful to distribute because it's going to prevent a lot of malaria.
So here's what I would do if I were running these phase 2 trials, okay? Phase 2 trials are done in people, but in a limited number, and they're all volunteers, and they're all healthy to begin with, and they've never had malaria before. So now what you want to know is, what is the lowest number of sporozoites you can inject into those people that have had this vaccination procedure and still get an infection.
Sure.
All right, so I still have a lot of work to do here.
You're rubbing your mic on your shirt collar. Sit up.
Up, or take the mic and move it.
It's fine. Beautiful.
All right. So the point is that we need to know more about how this vaccine works before we start going into the millions of doses.
What do we need to learn?
We need to know if you live in a high transmission zone or a low transmission zone, then how many sporozoites are present in the average mosquitoes in the high transmission zone versus low, and you can make some predictions then as about the efficacy of this vaccine.
This is going to take years.
What's the choice?
I'd like to start reducing malaria right away.
Well, they didn't reduce the malaria. They reduced the disease.
Okay, I'd like to start reducing disease. All right, you're absolutely right. In the discussion, we bring out important points.
What if it's $2 billion for the vaccine? Would you spend it?
If it prevents 100,000 clinical malaria cases? Sure. Bill Gates can do that.
What about the next time through, though? If you don't immunize again, what's going to happen to these kids?
These are very difficult questions. I don't have an answer.
Well, you have to eventually have an answer, though.
I see. These are the kinds of questions public health people deal with.
I know.
Is this ready to go? Should we spend the money? Should we develop it?
I know.
And the public says, no, let's get going here. We have to prevent kids from dying.
And the HIV groups were like that, too. I remember
Wow.
that. I do remember that. So it's a huge, huge problem.
I'm glad to be a basic scientist.
I'm glad I was a basic scientist.
You always are.
I think you never not become one of those. That's true. So these are not just theoretical questions that I'm asking right now. These are basic questions. Yeah, they are.
Anything else we need to mention before we move on?
Well, the other question that would arise, of course, is what branch of the immune system is protected here?
These studies don't address that.
They don't.
Because by looking at antibodies, all you can do is correlate them with protection, but
that's true.
You don't know if you don't look at anything else.
Yeah, so I have a question for the malariologists out there, because I know that there was a guy by the name of Anthony Cerami, and I know who he is too.
Tony Cerami.
Tony, that's right. He was at Rockefeller University, and his daughter went into science as well, and she got her. I don't know what she got her degree at NYU or not, but I know that she worked there for a while. And she was the one that identified the receptor on the endothelial cells of the sinusoids in the liver that the sporozoites attach to. So what if you could find out what that receptor was?
Yeah, you want to make a blocker?
Sure you do.
What if the blocker kills you?
Well, then you're dead meat, aren't you?
You don't use it.
No, you don't use it, but
you could make a blocker.
That would be a weird outcome, because I don't think that receptor is a functional receptor for anything else.
How do you know that? It's got to be there for a reason.
Not everything is there for a reason.
Yeah,
it could have been left over from a day gone by.
Yeah, I don't buy that. Everything's there for a reason. But you could make a blocker that may not interfere with its function.
The thinking was true for HIV virus.
Yeah,
like
but
that
it never worked.
No, it didn't.
Drugs are easier.
Yeah, well, it's easy to hop skipping around them too.
Blocking is I think, therapeutically more difficult than an enzymatic inhibitor, right?
Yeah. But if you had a temporary window of time, like, say, I'm thinking about now, visitors to the tropics, armies, if you had a blocker that worked and you didn't have to use it continuously, then you could still prevent malaria.
Maybe people are working on it.
But in little kids, it's not going to work when they live in the endemic areas.
Here's a population where you have 3 shots to get them something. Just 3.
See, that's right. That's just 3. Well, it's hard enough with just 1, isn't it? I mean, come on, your polio vaccine
1 is tough.
Just 1?
1 is no, it's 3.
Isn't it an oral 1?
3.
3 orals?
Yeah.
3 injections, 3 orals?
Mm-hmm. Yeah, and as you know, in India, you have to go back.
Okay, I learned something.
India, it's working. There's not much polio
I forgot
left.
that, actually. Yeah. I did forget that. So there are situations where you could apply this schema to the polio immunization schema.
WHO knows how to do that.
They do, of course they do.
They have the infrastructure for polio, so
they do.
They can do it.
What would be great to know in these infected, protected people is what is the level of parasitemia that was achieved, because every one of them was treated, by the way. They didn't let them go
after they became infected. Sure. I think that they will probably look at that and report it later.
Right.
And when they do, we'll do a TWiP on it.
Tremendous amount of work that still's out there, but I think they're to be congratulated. This is the first real good solid lead. So,
and you heard it first on TWiP. Well, not really.
No, not really.
But it's this week.
Every news agency in the world picked this one up.
Yeah, but this week we're doing it.
We are.
Let's do some email.
Hey.
This is from Chandler.
Chandler.
Hi, Doctors Racaniello and Despommier. After listening to your podcast on Onchocerca volvulus, I decided to contact you in hopes of enlisting your expertise. I'm a PhD student of biology at Northern Arizona University.
Know where that is?
And while my research is in novel antimicrobial gene discovery using halophilic archaea, I'm also currently working with Onchocerca lupi.
Oh, yeah.
There has been recently an increase in incidence of Onchocerca lupi infections in dogs in the southwestern U.S. My dog at 2 years of age began showing symptoms of an eye infection that persisted after several rounds of antibiotics. After seeking out an ophthalmologist, exploratory surgery was performed and a single nematode was found. Almost a year and 4 surgeries later, his infection is still present. The worms have been coming to the outside surface of his eyes where I see them and pull them out, as well as forming several nodules in his eyes. He's given monthly injections of ivermectin and takes doxycycline daily in hopes of killing the Wolbachia and rendering the parasites sterile.
Yeah.
Between only 2 vets I have seen in the Phoenix area, they have seen 9 cases of Onchocerca lupi recently. I am receiving nodules removed from the eyes of my dog, as well as parasite samples from California and Utah cases. I'm currently sequencing the partial mitochondrial genes from all samples in hopes of comparing the parasites in the U.S. to others published from Central Europe, where lupi is endemic. I wish to sequence the entire biome of Onchocerca lupi in hopes of discovering highly conserved unique DNA as a potential treatment target, as well as other possible endosymbionts as potential targets. That's cool.
Very.
I also would just like to see this information available to others who specialize in parasites. Funding, however, seems very difficult to secure for this parasite.
Yeah, that's true.
I have a couple of questions I hope you can answer, and if not, perhaps you can point me in the direction of someone who can.
I'll give it my best shot.
I have not found any literature on O. lupi that shows these subcutaneous nodules throughout a canine's body. The samples I've received have all been found in nodules on the eye. However, I am finding several bumps on my dog's body, and I'm wondering if these might be nodules or if they're a calcium buildup from the high long-term dose of oral prednisone. Secondly, after recently dissecting a newly formed eye nodule, I found several worms that I believe are calcified. I have not yet tested to be certain, and several worms that I am not sure if were alive or dead or perhaps unhealthy. Are there ways of testing whether these are healthy, living parasites? Will all dead parasites become calcified? Do you have any idea of why several of the worms have migrated to the outside surface of the eye?
Wow, those are a lot of questions, but I can begin by telling you that when nematodes, at least, that are parasitic die in the host, particularly tissue nematodes, they do calcify. Sure. And so one of them is called Filaria immitis. That's another filarial worm that's carried by mosquitoes now, and dogs throughout the United States are susceptible to this infection, and it's an endemic infection. And sometimes people get bitten by these mosquitoes, and they transmit the infection, but it never matures. It remains as an aberrant infection. And when they die, they're often misdiagnosed as something else. And so one of them is a coin lesion in the lungs. It looks like a little dime almost, with a little calcium rim. And that's a telltale sign for certain kinds of cancers. So it's misdiagnosed. And of course, when they do the sections on the tissue, after they remove it, they find that it was, Filaria immitis rather than a cancer, thank goodness. So the answer is to question 1, yes, most nematodes in the tissues will calcify once they die. Number 2, read that question again, W. Vince.
No problem.
Too long an answer.
Can you test whether they are healthy, living parasites?
Yeah, I mean, I don't know what kinds of biochemical tests you've got available to you, but certainly finding calcium deposits along the surface of the worm would indicate that it's dead. looking for intact structures inside the worm histologically tells you that at least if it's dead, it died recently. And then, of course, you can do viability tests. You can actually see whether the worms will move or not when you excise them from the tissues without damaging them. You can see them moving around so that they're alive. So those would be my basic ways of telling. Although you could isolate substances from the worms by just crushing it all up and seeing whether or not it's still there or not. Certain parasite structures deteriorate very fast once they die, and those would not give you a positive result this way.
Why would the worms migrate to the outside?
That's a good question, because I think the eye turns out to be an immunologically safe haven for a lot of parasites. Particularly the humors, okay, the anterior and posterior chambers represent a zone where antibodies don't usually go. Okay, and you can show this a lot with infections, again, in dogs, like Toxocara canis and things of this sort. The eye becomes a focal point, no pun intended, for these worms to actually crawl into. And once they're there, the immune pressure on them disappears, and they stay. Well, that's the basis for ocular larval migraines, because the worms then accumulate and start to damage lots of tissues in the eye. I presume that might be going on here too. As new infection is acquired over time, the old infections induce immunity all over the body, and the worm just feels so uncomfortable, it just keeps migrating until it gets to a place where it's comfortable. That's the best way I can put it, but I think that's
okay.
I don't think it's because they've been bitten in the eye itself.
I feel I had a million questions to ask you, and now can only remember these. I did want to point out that recently in areas where this parasite is endemic, Europe, a couple of papers have been published on lupi being zoonotic. These findings are based on molecular analysis. It seems like reason for concern in the U.S., where it looks like it is also soon to be endemic. So by zoonotic means it's in some animal.
Wild animals.
Yes.
And
it getting into the dogs that way.
So lupi indicates wolf, right? So lupus.
Yes.
That's right. So that's derived from that. So I think all dogs derive from wolves anyway, but maybe the wild wolf population has this infection as well.
I have included a picture from one of my dog's surgeries of what we are calling a drainage tract. It seems too large to be created for the adult worms to migrate through. Could it be for the microfilaria? Have you ever seen anything like this before? The vets and I are at a loss for what it is, but after having all surrounding tissue removed several months later, another one formed. Thank you for taking the time to read my ridiculously long email, and any information you can give me is greatly appreciated.
I'll take a look, but I used to do diagnostic work and stuff. Oh, I see what you're talking about there. Yeah, it's too big. Microflurry are even smaller than the adults, so I don't think that hole was made by. I have no idea what made that hole. And if the vets don't know, mostly, then nobody does, because they see a lot of stuff that regular physicians never get to view.
Yeah, it's pretty large.
It's very large.
You know any
no clue.
Anybody who would know? We could pass this on to them.
Well, the National Vet Service at
I
believe it's Iowa State University has the National Veterinary Pathology Laboratories there. Okay. That's where the original West Nile virus samples were sent.
Poor dog.
Yeah.
Next one's from Tommy.
Okay.
Hi, Vincent and Dick. Loving the podcast, and I'm glad you guys finally got around to the wonderfully peculiar and complex trematodes. Having worked with various different species of trematodes for the last few years, I'm glad to finally hear them make an appearance on TWiP. When you were discussing schistosome cercariae penetrating their host, I was reminded of some videos I filmed over a year ago, which can be found here, and he gives us a link
Excellent.
to that.
That's my pick of the week.
There, you will find the video of some cercariae of a fluke called Curturia australis, which I wrote a post about for the Parasite of the Day blog, and he gives a link to that. Penetrating the foot of its second intermediate host, a bivalve called Ostrovenus Stutchburii.
What's a bivalve, Vince?
It's something with two valves.
It's a mussel.
A mussel is a bivalve, but not a clam?
Sure.
They're all bivalves?
Yeah, they've got two shells.
Back on the subject of schistosomes, some recent research indicates that schistosomes can get divorced. Trends in parasitology. Male-biased sex ratio. Why and what consequences for the genus Schistosoma? and a few other papers he sends along as well.
Cool.
Also on the subject of trematodes in general, it is true that the majority of them have a snail first intermediate host, but there is a handful of families, gymnophalid, phelodistommatid, bucephalid, etc., which have bivalve first intermediate hosts. Finally, there are very few odd species which use polychaetes of all things as first intermediate hosts.
Cool. Very cool.
Cardicola forsteri, however, such examples are very rare. So generally speaking, all Dignian trematodes have mollusk first intermediate hosts, and the majority of them have snail first intermediate hosts.
Okay.
Dixon is giving you the thumbs up.
You bet.
Keep up the good work. The podcast is always informative, and I look forward to more fluke talk.
Well, you're going to get some.
And the last one is from Kenton, who writes, are there hieroglyphics indicating schistosomiasis was a recognized disease in ancient Egypt? Thank you. I enjoy listening to your podcasts on TWiP.
Thank you very much for the question, too. Actually, there claims to be some indicating that men at a young age, let's say in their teens or even preteens, exhibit bloody urine.
Yes.
Bloody urine
schistosomiasis,
in women.
Hematobium.
That's right. Ooh, Vince. Nice going.
I remember that.
Nice. Women, as they reach menstrual cycle age, puberty, begin to menstruate, and they have blood in their. It looks like they have blood in their. It's not in the urine, but
it mixes in. Sure,
it mixes in. Will men, they thought in Egypt, have the same syndrome? But that really indicated to me that it was schistosoma hematobium that they had acquired. In fact, both males and females would have acquired it, so you couldn't distinguish between menstrual cycle bleeding and bloody urine from schistosoma hematobium in women either. But I think the hieroglyphs show male bleeding.
So I found an article by Gina Colata.
Oh, yeah. She writes for the Times a lot.
There is a hieroglyph showing a bleeding penis, which is a symbol for schistosomiasis.
There it is.
See?
So that could be your pick of the week. How's that?
The schistosomiasis symbol is pronounced AAA.
That's how you feel.
The Ebers Papyrus of about 1500 BC. So there you go.
Nice.
We'll put a link to this for you.
Yep.
In the show notes.
The Egyptians were observant at every level.
Why do you think that was?
Well, because they lived in one place for a very, very, very long time. And they could describe what went on there extremely well because they were highly familiar with it.
Egyptians were amazing.
You got it right.
They came and went, though.
Well, it took them a long time to went.
Yeah, they did.
And they're still here, by the way. Don't say
there
that
are
they
Egyptians.
came and went.
We're talking about the ancient Egyptians.
Oh, well, every ancient civilization's gone now.
We talked about this once on TWiP, and one of our listeners in Egypt said, there's still Egyptians around.
That's right.
Okay.
Don't write us off yet, especially after the Egyptian spring.
Dixon, where can you find TWiP?
TWiP. Well, you can find it on a lot of different places.
That's right. Like iTunes.
Microbe World.
Microbeworld.org/TWiP.
You can find it there, iTunes.
And if you want to...
You can find it online if you want.
Stream it to your iPhone or Android phone, you can go to microbeworld.org/app and get an app for that. Yep. And if you want to ask us a question, people should send it to twip@twiv.tv. We love your questions. We have plenty more lined up. We haven't done this for a while, but Dixon and I are going to make a New Year's resolution before the new year begins to try and do it more frequently. Right, Dixon?
You bet.
Thank you, Dixon, for doing this today.
Vince,
I know you're off to China.
My pleasure.
You're a busy guy.
We're all busy.
I like talking to you.
And I like listening to you.
Well, I like listening to you, too.
No, I like the interchange. It's not only taxing with regards to trying to dredge up these answers, to be honest with you. And so this requires that we sit down and think about these problems a lot more than we do ordinarily, which is a good reason for doing these, by the way.
Yeah.
But it also means that we have the same interests because there's no difference in the way you think about a virus or a bacteria or a schistosome. They have all got the same goal in mind, and that is to take advantage of you.
Absolutely.
So how do they do that? And that's what this show's all about.
Dixon Despommier is at trichinella.org, verticalfarm.com, and medicalecology.com. I'm Vincent Racaniello. You can find me at virology.ws. You have been listening to This Week in Parasitism. Thanks for joining us. We'll be back next time. Another TWiP is parasitic. you.